Okay, hello. Um, in this video, I'm essentially just going to be going over a couple of articles relating to biogerontology that I found online in the Nature Aging Journal, and going over some notes. I am not an expert or a professional. I am just interested, and I'm going to be, I guess, sharing the article. So this article is called Spatial Mapping and Senolytic Targeting of Senescent and Disease-Associated Microglia in Aged Mouse Brain White Matter, which sounds complicated, but essentially what it is spatial mapping is just a type of analytic technique where they visualize the organization of molecules in the tissue. So they use something called high throughput techniques to detect distribution of biomolecules, which essentially is them using things like microscopy to look at how the molecules interact with each other in a more holistic way. And oftentimes they will use automatic equipment to test many different things, such as the cell-to-cell interactions, the effects of different chemicals and compounds on the cells, by just rapidly going through many different compounds and waiting until one of them has an effect. And so this is just a way for them to essentially look at how the cells interact in the tissue. Now, senolytics is probably not a term you're going to see outside of biogerontology very often, but senolytic is a certain type of cell or molecule that has the ability to kill off senescent cells. And senescent cells are cells that are going through the process of biological aging and eventually cell death. So specifically, senescent cells are a type of cell that is approaching the end of its lifespan and begins to express certain age-related genes that cause illnesses, and specifically, and most importantly, currently in scientific literature and research, inflammation. As of right now, most biogerontology research that I've seen, at least, focuses on the immune system and its interactions with inflammation in the body, because inflammation is the cause of most of the symptoms and long-term damage that you see in senescent cells. These senolytics are used oftentimes as a way to just kill those senescent cells off so that the only cells left over are the healthier non-senescent cells that are not currently undergoing the aging process, which will reduce your inflammation. Which is I need to stop saying I'm trying to do that. Which will make it easier for your body to fight off infection and to not have as many chronic illnesses and pains, and to not be as at risk for things like different bone-related and muscle-related diseases. So it's just generally a very good thing to not have inflammation. Microglia is a type of cell in the brain. It is a type of macrophage, and a macrophage is a white blood cell, so it's an immune cell. This is a type of immune cell in the brain, specifically in white brain matter, that goes and does immune cell things. Specifically, it's a type of glial cell. You probably don't need to know what that is. It's not really that important. But it's a specific type of cell in the brain and the spine. And it is the white blood cell specifically for the brain. So what they found in this study, and I'm going to read it all out, right? The whole point of this video is I'm going to read it. But the main thing about this study is they found that these microglia will eventually begin, as they undergo senescence in the white brain matter next to the hippocampus, they will eventually start to express different disease-related genes, but they will also go and they will cause other cells to trigger their own senescence. So they will go out into the brain and they will cause other cells to begin the process of undergoing senescence. And this will more rapidly lead to inflammation, and specifically this gene which expresses a protein called lectin 3 that is a known cause for inflammation and other cardiovascular and uh, immune diseases. So the abstract is: white brain matter undergoes structural and functional alterations linked to late life cognitive decline, but the cellular and molecular basis of its selective vulnerability remains incompletely defined. Here, in naturally aged mice, we demonstrate that senescent and disease-associated microglia, DAM phenotypes, converge in hippocampal-adjacent white matter. So this is white matter next to the hippocampus, which is a part of your brain. And phenotypes is essentially just saying that it is expressing a certain type of physical characteristic, which is caused by the genes that it is expressing. Particularly in the fimbria, which is close to the hippocampus, using regional gene expression profiling, immunolabeling, blah blah blah. You don't need to know what any of this is. We identified an aged brain-exclusive microglial population concentrated in white matter that expresses DAM genes together with a sen-brain senescence gene signature, including galectin 3. This is the protein I was talking about. I might have mispronounced it earlier, but it's called galectin 3. And you can look it up yourself, but it causes some types of inflammation and other diseases related to your immune system and your cardiovascular system. Excuse me. And this microglial population, that's so hard to say, microglial population in white matter will cause this. And these analyses they've done with the spatial mapping have shown that cell fate transitions may give rise to this senescent and DAM-like state. Now, a cell fate is just the type of cell. So, cell fates is essentially just describing a process through which cells that are originally not specialized in anything will transform into a specialized cell. So, for example, a non-specialized cell might transform into a blood cell. A non-specialized cell might transform into a neuron. A non-specialized cell, as the body develops during aging, might transform into a liver cell. Excuse me. And what these microglia are doing they are changing the cell fates, or the type of cell that these non-specialized cells are transitioning into. So instead of transitioning into a regular neuron, this cell that was otherwise functioning regularly might transform into a senescent neuron, or it might transform into something that is not a neuron. It might transition into an aged pro-inflammatory senescent state. This is what is being caused, right? It is transforming these cells that were otherwise healthy into senescent inflammatory cells. And this is essentially just saying that there might be a link between the microglia and this pro-inflammatory state in the brain. Pharmacogenetic or pharmacological senotherapeutic interventions reduced white matter Gal3 DAM abundance and restored a more youthful microglial organization in aged fimbria. These findings identify a senescence and DAM-enriched microglial state as a prominent and partially reversible feature of aged brain white matter. So, like I said before, I'm not an expert in any of this stuff, so you should take everything I say with a grain of salt. And the point of this video is that I'm going to be reading it and learning this information along with you. So don't treat me like an expert. Treat me like someone, treat me like like a friend of yours who just found out about this and is trying to explain it to you. Okay? So it seems like this state that is being underwent by these other cells, which is caused by the microglia, is a prominent thing in aged white brain matter. Aging increases the risk of cognitive decline and neurodegenerative conditions, yet the cascades of cellular and molecular events that confer region-specific brain dysfunction in aging remains unclear. The hippocampal formation is an age-vulnerable mediator of learning and memory, compromising a cortical gray matter infolding directly abutted by dense white matter tracts. Abutted just means it's next to. The corpus callosum and fimbria fornix, I don't know what those are either, it doesn't matter, as core components of the Papez circuit. The hippocampus and fimbria fornix transmit signals to the hypothalamus, thalamus, and cingulate gyrus, supporting the memory and emotion-related functions. So these parts of the brain support memory and emotion, and learning and other similar things. Imaging studies reveal that white matter dysfunction, and particularly changes in the fimbria fornix, may precede cortical gray matter dysfunction. So this change in microglia may precede changes in gray matter, which can cause dysfunction, or disease, or illness, or inflammation, such and such. So this is like this change in the microglia is causing further changes down the line and acting as a sort of, calling it a cascade would not probably be scientifically correct, but you can think of it like an avalanche, where it starts with a little bit of snow, and then it begins causing everything else in the brain to also undergo this sort of senescence. Well, saying everything else in the brain also isn't correct. Gray matter, I should be specific. It's causing gray matter to become dysfunctional. This represents an early corticolimbic impairment underlying memory dysfunction and the progression of cognitive decline. So, like dementia, for example, might have a relationship with this microglia. Cell-specific molecular mediators of aging. Cell-specific molecular mediators of aging, okay, are potential intervention opportunities for preserving brain health and preventing neuropathogenesis in aging. Cell-specific molecular mediators. Cell-specific molecular mediators of aging. What is a mediator of aging? Oh, they're talking about the senolytics. These are the senolytics. So the things that can kill the senescent cells. Preserving brain health, preventing neuropathogenesis, okay. Inflammatory and senescent fates have emerged as, so these senescent fates are the cell types, have emerged as maladaptive cellular trajectories in aging tissues. In the aged and neurodegenerative brain, DAM accumulate and exhibit downregulation of homeostatic genes. In the aged and neurodegenerative brain, DAM accumulate and exhibit downregulation. Downregulation means that genes are being expressed less often of homeostatic genes. Let's look at the word homeostatic, because I don't know what that means. Oh, I see, homeostasis. Okay. So it is downregulating genes that cause homeostasis. And homeostasis is just the cell being able to keep its insides separate from its outsides. Like, for example, your body is capable of keeping your heat at a certain level separate from the outside world. That is homeostasis. Your cells keeping a different concentration of water than the outside of the cell is homeostasis. Different ions, like salt. You only want a specific amount of salt inside of your cell. That's homeostasis. You only want, you definitely want a barrier between the inside of your cell and the outside of your cell. If your cell has a cell wall or a cell membrane and that gets punctured, then the insides of your cell go outside, and that's really bad because the cell essentially just instantly dies. This is actually what causes soap to kill bacteria, is soap and chlorine as well puncture holes into the cell membranes and walls of bacteria and cause the insides to go outside. and that, is pretty bad. They die from that. So that's what causes them to be effective. It's why we use chlorine in our pools and why we use hand soap on our hands and so on and so forth. So it downregulates these genes that cause homeostasis alongside upregulation of neuroinflammatory, lysosomal stress, and neurodegeneration related genes. So it causes genes to be more active and more used that cause inflammation, stress, neurodegeneration, that sort of thing. The whatever this is called pathway represents a critical regulator of dam-like microglia, which are thought to initially confer protective functions before shifting to a deleterious fate, including inflammatory disruption of brain microenvironments. I'll be honest, I don't know what that means. I don't know what this pathway is. This is the first time they're mentioning it. What is the apoTREM2 pathway? The TREM2 apo pathway drives the transcriptional phenotype of dysfunctional microglia in neurodegenerative diseases. Apolyproprotein E. I have no fucking idea what that is. Form models of that. It mediated a switch from a homeostatic to a neurodegenerative microglia phenotype. I see. So this is like a light switch that causes, you flick it and it causes the microglia to switch from regular microglia to degenerative microglia. Okay. This pathway is a critical regulator. I see. So it's a light switch. You flick it on and off and it causes the change in cell fate. White matter microglia fulfill overlapping and distinct roles relative to microglia in gray matter. White matter microglia, blah 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 blah. Gray matter. Okay, gray matter and white matter microglia do similar things, as defined by interactions with densely myelinated fiber tracts and neurons, respectively. Single-cell RNA sequencing identified white matter-associated microglia as a TREM2 dependent population that increases with aging. Okay. It is dependent on this pathway, and it increases with age, which makes sense. This study and others collectively demonstrated that WAM are enriched for molecular signatures related to lysosomal function, phagocytosis, iron regulation, and lipid metabolism that do not clearly separate from DAM. Thus, we ascribe the white matter designation as an anatomical characterization and use the broader DAM designation to refer to the molecular profile. Okay, so they're saying that from what I'm understanding of this, I could be wrong. They're saying that these specific microglia are overlapping with the DAM microglia. The disease-related microglia have similar functions and similarities to the regular ones, and that even in the DAM state, they still have some functions related to regulation of the immune system. And so you cannot just say that they're completely separate because they have a lot of overlap with regular microglia. So you can't just immediately look at a population and say, oh, this one's DAM and this one's the other one, because they overlap quite a bit. Characterizations of the disease-related microglia from aged and Alzheimer's disease brains have revealed overlapping phenotypes with senescence. Senescent DAM exhibit gene expression profiles that include silin-dependent kinase inhibitors, chemoattractant factors, pro-survival factors, canonical dam factors, and lysosomal stress factors, encoded by LGAL3, which binds to B-galactosides and B-galactosidase on stressed lysosomes, and is an established microbial factor that interacts with neuroinflammatory receptors, contributes to phagocytic remodeling, and is linked to maladaptation and disease states. Okay, so they're just showing us the connection between the specific gene that is being expressed in these dam microglia and the protein that is being produced that causes this neuroinflammatory state, and it contributes to all these different diseases. They're saying this dam expresses this gene, and this gene makes this protein, and this protein causes bad stuff. Senescence-associated B-galactosidase activity is widely used lysosomal stress senescence biomarker, okay, including insenescent dam that expresses blah blah blah. We propose GAL3 is a senescent dam biomarker that requires further characterization by spatial mapping. So they're saying this specific gene / protein is specifically very important when it comes to knowing whether or not a cell has become senescent or has become more inflammatory. And that there needs to be further study of the specifics of what causes that and why it is a good biomarker. Emerging spatial omic technologies permit high-dimensional mapping of cellular and molecular states with preserved anatomical tissue context. Emerging spatial omic technologies. Man, how is anyone who isn't a specialist in the field supposed to know what this stuff is? Permit high-dimensional mapping. High-dimensional mapping. Sci-fi crap. Of cellular and molecular states with preserved anatomical tissue context. That seems like a really complex way of saying that it is a very in-depth way of looking at the cell. These technologies map the cellular states and preserve the context of those cell states within the anatomy of the tissue. The anatomy of the tissue. So it's saying not only do we know where these cells are relative to each other, but we also know where they are and their states relative to the overall tissue. We know specifically where it is within the brain. We know specifically where it is compared to the other cells around it. And we know where it is compared to whatever specific tissue in the brain we're looking at. These platforms overcome isolation artifacts and population biases inherent to dissociative single-cell methods and facilitate classification of rare cell populations. These platforms overcome isolation artifacts and population biases inherent to dissociative single-cell methods and facilitate classification of rare cell populations. Man, I don't know how you're supposed to know what that means. Unique phenotypes and cell-cell communication. These platforms overcome isolation artifacts and population biases inherent to this phenotypes and cell-cell communication. Okay, so they're saying that it is a more broad way of observing the cell, and that it is capable of picking up on many different types of unique interactions and phenotypes and different types of communication that you would otherwise not be able to recognize with a more specific or less holistic observation method of the cells that we're looking at. So these microglia come in and they do some unique stuff that we haven't seen before, and they come in and they start sharing different types of cell-to-cell communication we haven't seen before. We would be able to notice and recognize that from the information we receive from our specific technologies and spatial omic techniques. Importantly, 2 recent studies of aged brain cell signatures analyzed by spatial transcriptomics identified white matter as distinctly altered in aging. Okay, white matter undergoes a big change during the aging process from previous studies. Building on our previous work Typifying aged microglia in single-cell workflows, we sought to investigate senescent and damn microglial identities in age-vulnerable corticolimbic gray and white matter naturally aged mice. Okay, purpose of the study. Through use of 3 spatial mapping platforms, they're telling you how they're doing the spatial mapping. You can look that up if you want. I'm not going to because I don't care. Together with gold standard gene expression and imaging techniques, we mapped senescence in damn cell features, which exhibited pronounced accumulation in aged limbic white matter. We delineate a white matter enriched microglial population that accumulates with age and is characterized by the highest expression of the sen brain gene signature, with particular enrichment of senescence associated secretory phenotype. Organelle stress. Holy shit! What a long sentence. That is one sentence. We delineate a white matter enriched microglial population that delineate. Describe. We are describing a white matter enriched microglial population that accumulates with age and is characterized by the highest expression of the sen brain gene signature, with particular enrichment of senescence associated secretory phenotype, organelle stress, and cell surface domains. Okay, they're just telling you what type of cell they're looking at. Bioinformatic trajectory inference demonstrates multiple possible transition states involving age-increased microglial populations. Okay, so there's multiple different types of cells that they can turn into, also harboring molecular senescence and damn biomarkers, and these different types of cells also have the biomarkers related to inflammation, aging, and senescence that terminate in the old age exclusive inflammatory SASP enriched population, senescence associated secretory phenotype, that terminate in the old age exclusive inflammatory phenotype enriched population. We applied systematic pharmacogenetic and pharmacological strategies to target senescent cells, which reduced aged white matter senescence and damn biomarkers and restored microglial morphological and organizational properties in the aged fimbria to a more youthful state. So they were capable of going in and using specific strategies that they've used before to target these microglial senescent cells and the other cell fates which are caused by the microglia, and reduced the specific markers and biomarkers they found in the study and restored properties in the fimbria to before this senescent state. So before we go to results, let's do a review. So far, they found microglia in the brain that cause senescence and have the specific gene that causes senescence and inflammation. And they found that it has a lot of overlap with gray matter. They found that it has a lot of overlap with regular microglia, and that just generally in this area, there's a lot of overlap between the different types of microglia and the functions that they perform. They also found that these microglia can cause cells to go into different cell fates, which all share this same property of expressing senescence-related genes and proteins, specifically at GAL3. They are using techniques in the cell to look at the holistic tissue and cell interactions, and using specific techniques within these senescence-enriched gene populations, senescence-related populations of cells, to target them and cause them to revert back into a non-senescent state. So, we previously demonstrated that the aged female hippocampal regions harbor increased DAM and senescence markers. We investigated senescence and microglial gene expression patterns in microdissected Fimbria fornix and corpus callosum white matter relative to hippocampal gray matter of aged female and male mice. We distinguish between detection of this and that. Man, do you really want me to look up what all these are? I'm not going to do that. Expression were significantly higher in fimbria fornix and corpus callosum compared with hippocampus. Okay. So in the specific type of white matter next to the hippocampus, it's much higher than in the hippocampus itself. And specifically in female mice, it seems to be happening faster or to a larger degree. Expression in female mice was significantly higher in the fimbria fornix and corpus callosum compared with the hippocampus. Yeah. Relative to young counterparts. This expression increased in both gray and white matter microdissections of old male and female mice. If I knew what these were, that might mean something. I'm assuming TREM is similar to that pathway we looked at before that is that light switch that causes the microglia to turn into the senescence-related microglia. So this being expressed more in the older counterparts is essentially saying that these genes are more expressed in the older mice than in the younger mice, which may be related to these pathways we've looked at before in the study that have been shown to cause senescence. So these genes were more expressed in old female and male fimbria fornix and corpus callosum. And so all these different genes, they're essentially saying the same thing like 3 different times. There's a bunch of different genes related to senescence pathways that are more upregulated in older populations of mice than in younger ones, consistently. And it is more upregulated in the fimbria fornix and corpus callosum than in the hippocampus or cerebellum. Which means that the white matter is susceptible to late life changes in senescence and DAM transcriptional signatures. So white matter is more malleable for being turned into this type of specific senescent state. You can go over this yourself if you want to. I might just take a look at it. Since I don't know what any of these genes do, I would have to look them up and then come back here, look at the graph, look it up, look at the graph over and over again, which would make this video like 50 minutes long, so I'm not going to do that. You can read this though. P-values were significant. Okay. So they found there was a significant difference between the old and the young mice in terms of the expression of these disease-related genes. Gal3 myeloid cells accumulate in the hippocampal adjacent white matter of old mice. Based on the observation that senescent DAM cells express Gal3, we investigated the abundance of Gal3 immunointeractive cells across the hippocampus and adjacent white matter in young and old brain sections. Gal3 cells were rare in young mouse brains. In aged brains, we discovered Gal3 cells in multiple limbic regions, with dense localization in white matter tracts, especially the fimbria. In aged fimbria, Gal3 signal colocalized with the myeloid microglial marker, colocalized. Okay. So they found Gal3 with microscopy to be in the same cells that had the myeloid slash microglial marker, but not with the astrocyte marker, which says that this microglial cell is usually in the same place as this Gal3 protein which is being expressed, which leads us to believe further that the microglia are the ones producing the Gal3, which is inflammatory. Gal3 immunoreactivity compromised a larger area of old versus young female fibria. However, Gal3 area was not significantly different in fibria between old and young male mice. Oh, that's interesting. Gal3 immunoreactivity comprised young female fibria. However, Gal3 area was not significantly different in fibria between old and young male mice. So there is quite a big difference between the Gal3 expression in female mice compared to male ones. So this specific microglia may activate in a more extreme way in one gender than the other, which is interesting. In the old female fibria, Gal3 cells were larger relative to Gal3 plus cells were larger relative to Gal3 minus cells. I don't know what that means. Populations from old mouse fibria revealed size-specific principal components based on cellular morphology and distinguished by Gal3 plus immunoreactivity. So there are specific parts of the cell that are components of the cell that are size specific that have immunoreactivity to Gal3 plus. So there are specific types of the cell that will react to Gal3 plus, but only if the cell is of a certain size. Otherwise, it won't have that. And what did they say earlier? That in the female ones, these Gal3 plus cells were larger relative to the minus cells. Interesting. Okay. Similar to the fimbria, anterior commissure and corpus callosum white matter tracts exhibited strong Gal3 plus immunoreactivity. EBA1 and Gal3 also colocalized in deep cerebellar nuclei. So these are next to each other, or expressed with one another. Basically everywhere that they found in the study, or that they looked at in the study. Thus, immunofluorescent imaging and gene expression demonstrate increased abundance of the senescent and dam marker Gal3 in aged white matter, including the fimbria, which was more pronounced in aged female mice. This is the one thing to take away here, is that this specific senescent protein was found significantly more in aged female mice and in aged male mice, but to a lesser degree. IMC demonstrates microglia positive for senescent and dam markers are abundant in the aged fimbria. To investigate the molecular phenotypes of aged microglia across microenvironments, we implemented IMC, which combines immunolabeling and time-of-flight mass spectrometry to map and quantify 10 protein markers with 1 micrometer spatial resolution. Micrometer? micrometer spatial resolution in the aged female mouse brain. So, time of flight mass spectrometry. I don't know what time of flight mass spectrometry is, but they're using immunolabeling. I'm assuming labeling is just like a form of fluorescent labeling, and the mass spectrometer to map and quantify 10 specific proteins or protein markers with a really small spatial resolution in mouse brains. So they're looking at 10 proteins. We measured the abundance and colocalization, so if they were found together, of senescent and DAM markers, these. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So these are the 10 markers that they're talking about. Fimbria myeloid cells exhibited greater intensity of these markers relative to hippocampus, which exhibited greater this type of protein level. Stratification of Gal3 plus and Gal3 minus cells in the fimbria show that the Gal3 plus cells harbor trending or significantly higher levels of senescence-related markers, UPAR and CD38. So, specifically, this form of the protein is really important for senescence. This is the one that is causing the majority of the senescence-related disease and inflammation that you're seeing in these cells. These protein-based results validate and extend our observations from regional gene expression and immunofluorescent analyses, demonstrating that the aged fimbria accumulate senescent and DAM markers, including Gal3 and UPAR. Now, what is IMC? We should look at that. IMC spatial mapping in biology. I guess we would need to know what cytometry is first. Measures the number and characteristics of cells. I just looked at this. Why is it so hard with all of these different scientific disciplines to just figure out how something works? Like, I've got to go to a specific journal article just to figure out what cytometry is and how it works. Why? This should be in the Wikipedia article. Why is the mechanism not in the article? AI generated? No, fuck off. Here we go. In flow cytometry, cells are focused into a narrow stream and carried in a single file in a sheath fluid past a variable number of lasers. Light scattering and fluorescence is detected by a series of photomultipliers. Forward and side scatter of the laser light gives information about cell size and morphology. Different cell components can be labeled with antibodies bound to a fluorophore, so same thing as fluorescence microscopy. And the fluorescence measured at defined wavelengths to provide information. Nucleic acid fluorescent stains are used. Okay, so it's a combination of multiple different methods which looks at the holistic characteristics of a cell. I see. In the solid phase, it's filtered through a membrane, labeled with a fluorophore, and scanned by a laser. I see. Okay, cool. Interesting. How much more do we have here? Just highlight that real quick. Oh my god. I might have to split this up. Wow, why is there so much? No, we might just skip the results, I'm gonna be honest. The results, honestly, usually the least important part of the paper anyways, unless you already know what they're talking about. Since we're not experts, we don't really know what they're talking about. So if you want to go in here and figure out what all of this means, you can, I guess. You could analyze if the study is trustworthy or not off the results if you do know. But honestly speaking, the amount of time that would take would probably not be worth it. We'll probably gradually learn what a lot of this stuff is just by going over other studies as well. So I would recommend not to. Probably a waste of time. Let's just go to the discussion. Well, I guess we can read the last part of the results. Okay, discussion. White matter integrity is a key determinant of neuronal activity and cognitive function. In mice and humans, white matter tracts are selectively vulnerable in aging, and maladaptive microglia mediated neuroinflammation is emerging as an underlying driver. We applied imaging and gene expression analysis with spatial molecular profiling to map cell type specific morphological, anatomical, and molecular features of microglia in the aged mouse brain, with a focus on signatures such as proteins and markers and so on. Blah, blah, blah. Mm-hmm. Gal3 plus again is important. It is more common in white matter than in gray matter. You know what? I might just do that. Instead of reading it directly, I might just summarize from now on. Okay, the Fimbria specifically had more senescence proteins. UPAR, a cell surface senescence marker under immunotherapeutic development, is highly expressed in aged white matter Gal3 plus cells, highlighting a potential alternative targeting strategy. Interesting. So, Gal3 plus, they're saying here, is very different from Gal3 minus. Gal3 plus is the one you want to look out for. Gal3 plus is the one that is causing the majority of the disease related expression of biomarkers and stuff. Right? This is the one that's causing the issues. Okay, so they're recognizing a couple of limitations of their technology, that there might be extra detection of these biomarkers um, from adjacent cells rather than the actual cell they're targeting. Targeting of P16 reduces senescence and DAM markers in aged white matter. Interesting. Oh, interesting. What is this notion? The fimbria is spatially adjoined by choroid plexus in the lateral ventricle, heightening its exposure to circulating factors that infiltrate immune cells. Okay. So they're saying these senolytic strategies would be capable of targeting these age-increased phenotypes in the microglia and other cell fates, and it is likely that it would target other cell types. Related to this, this area specifically that is having this increased upregulation of the bad phenotype is right next to a part of the body that is exposed to uh, different factors that can infiltrate immune cells, such as the microglia. So there is a potential area here they could target where they could place these senolytics next to the fimbria and have it circulate throughout the body and have it infiltrate these microglia in the fimbria, um, as a type of therapy that would potentially reduce all of this senescence. So they also need to be careful that using these senolytics does not somehow directly affect the integrity of white matter as you age. They do not know currently the full effect of how these senolytics affect the cells in your body. So they know that it has a negative effect on senescent cells, which is good because you want to get rid of those senescent inflammation causing cells, disease causing cells. But you also don't know fully how it's going to affect the specific tissues in every area of your body. And so if you just use these wantonly, you're not going to fully understand what it's going to do to your white matter, as it alters these microglia, and it might cause issues further down the line. Okay, so in this one, they're telling us how these senescent microglia may come into existence. They might start expressing this bad gene after migrating to white or gray matter niches and obtaining a new phenotype. So persistent stress in anatomical regions may cause the change. They also may get signals from blood and CSF to activate that phenotype. And the ones in the white brain matter may be more impacted by the signals derived from the blood and CSF than from the other two. I'm not exactly sure what they mean by stress. Are they talking about like stress as you would typically think of stress, or are they talking about physical stress? Are they talking about football athletes who get hit in the head and get damaged brain matter, or are they talking about people who undergo emotional stress? I'm not sure. In science, stress is more loosey goosey than in like psychology. In psychology, stress is very clearly defined as like, you go to work and you work for 8 hours at a desk and now you're stressed out. Whereas in science, stress is more like anything that would cause stress or damage. I don't know what a synonym for stress would be, but for example, getting hit by a bowling ball would stress your abs, you know? Uh, Getting shot with a gun would stress your immune system because now you have an open wound. And also your circulatory system would be stressed by exercise. And different types of stress can be good and some can be bad. Like there's a certain type of stress from exercise that you can get that positively affects your muscles, for example. So stress is not necessarily always a negative thing. And in this situation, I would really like if they were more specific with what type of stress they're talking about. I think they don't really know yet. I'm assuming they're just saying that stress in general may be a cause, specifically for white brain matter. We don't know what type it is yet that causes it, but maybe stress in general just from aging. Who knows? But persistent stress may contribute to that, as well as signals from blood and CSF. And then migration from the white matter to gray matter may cause this damn phenotype. This damn phenotype. And the prominence of each may correspond to whatever specific subregion of the brain you're in. But, okay. I see. So this might be one of the forms of stress they're talking about here, which is lipid-rich, which is fat. Fat-rich debris can overload the lysosomal system, triggering lysosomal membrane permeabilization, which means that the membrane, things can move through it more easily. So, lysosomal, let's look at that real quick. Lysosomal membrane. Lysosomal. Well, let's flip that. Oh, now I remember. It's an organelle. I see. Protects from degradative enzymes within the lysosome. So, fat-rich debris can overload the system, triggering the membrane to become permeable, which leads to the accumulation of Gal3 protein. I'm assuming the lysosome has some sort of hand in the cell producing Gal3, and this allows for the Gal3 to leave the lysosome and enter into the cell. I could be wrong about that, but that's what I think they're saying. An overburdened lysosomal system may shift DAM from an adaptive program into a senescent maladaptive state. So stress to the lysosomal organelle and the lysosomal system may cause this shift into a senescent state for microglia. So I'm going to write that down. Lysosomal stress may cause shift from normal to DAM/senescent state in microglia. Okay. Senescent cells display increased lysosomal mass, altered pH, and lysosomal damage. Interesting. Okay. Aged increased white matter microglial Gal3 signal may reflect chronic phagocytic load and cumulative lysosomal stress associated with persistent myelin and debris clearance in the aging brain. Interesting. That is so cool. So, so they've already distinguished a certain type of stress that may be causing it. So as you age, there's an accumulated stress to the organelles, the lysosomes within your microglia that cause them to be less efficient at keeping the enzymes that they produce separate from the rest of the cell. This Gal3 exits the lysosome and enters into the microglial cell as a whole and causes it to shift from a normal state into a maladaptive senescent state. And then this causes an avalanche of changes where that microglia will then go out and shift to other parts of the brain matter, and specifically the gray brain matter next to the hippocampus, and will then cause other cells to exhibit different cell fates similar to that microglia, which will cause them to become senescent or maladaptive themselves. So, very interesting. Very interesting. That's cool. They showed that expressing DAM in juvenile brains is irreversible, whereas neonatal DAM is plastic. Interesting. So if this phenotype is expressed in the brain at an extremely early age, the brain can actually adapt to it. But after a certain age, the brain no longer becomes capable of that. So that suggests that this is probably an evolutionary change that we have, like an evolutionary what is it called? An evolutionary trait that we possess that causes us to form these damn states after a certain age for some unknown reason. Maybe it helps people reproduce. Maybe it's just a natural thing from aging. That's interesting. A separate study demonstrated that microglia from 24-month-old mice harboring that and senescent markers are resistant to microglial depletion and are associated with recruitment of T cells, myelin perturbation, and axon degeneration. Interesting. So once you start it, it will continue even if you try to remove the microglial cells. They will replenish and continue to recruit T cells that will degenerate your neurons. So it's like a switch has been flipped, and these microglia in your brain are now in this state essentially permanently past a certain age and will continue to degenerate your brain consistently, even if you try to remove them. So they have found a way to reduce this, to reduce this while still preserving cognitive function in mice through a therapy. So they've found that other parts of the body also express L-gal3 and other senescence markers, such as in the spinal cord. Interesting. Okay. So these specific microglia in the brain matter cause these cascades, which eventually lead to cognitive decline. That's very interesting. I like how much overlap there is between biogerontology and um, these type of neurodegenerative and other degenerative diseases such as Alzheimer's and dementia. There's a lot of overlap here between them, and I find that very interesting. Targeting of these genes is an effective strategy to counter DAM and senescence-linked profiles and reverse microglial molecular and cellular organization into a more youthful state. Cool. Let's do a quick skim of this. We're not going to go over it super thoroughly. Da-ba-da. Da-da-da-da. How many mice did you use? Um, how many mice were used? Do they not tell us how many mice they used in their experiment? Let's see. Research mice. They just say a population, but they don't say how many. Mouse treatment sample sizes were 5 to 8 mice per group. Oh, that's tiny. Per group, but how many total? Okay, they're similar to previous publications, so that's good at least. Excuse me. Four HT1 mice. So it looks like their sample size for the total amount of mice for each group that they did experiments on was around 5 to 8. I'm not sure how many groups they had. Assuming they had at least a couple. At least At least 3. Probably more. Still, even if there are many groups, the fact that there's only 5 per group is pretty bad. So, not great. Still really cool research. I do wish the sample size was bigger though. Like, I'm not asking for 100 mice or anything, but there should be at least 10. Only 4 or 5 is... It's just not good. Man, I feel like it should be easier to find how many mice you used. This should be something you just state somewhere in the study, right? Like, I really shouldn't have to dig around the study to find how many mice you used. You should just place this at the beginning of the methods. Animals. This many mouses, animals, methods. First sentence. 100 mice were used in this experiment. Period. Everything else, right? Like, this should be like the first thing you say. Um, I understand, you know, I haven't written more than like 1 or 2 really shitty amateur journals for college. I don't know how your journal wants you to write these. I don't know how you were taught. I don't know what the correct way of doing it is. But I feel like, as a layman, this should be like the first thing you say in any journal, in any article, right? This is the sample size, period, everything else, right? The fact that I have to dig for the sample size is crazy. Why do I have to go through all of this information that I don't need on specific methods and testing and stuff just to figure out what the sample size was? N equals 5 mice, so sample size is 5 mice, 2 to 3 sections per mouse, maybe. But for this specific testing, so I don't know if that's for all of them. 5 to 8 mice per group. I don't know, man. That's just not a lot. It's okay, I guess. It's not awful. Um, let's see. Biological study. I probably could have worded that better. Forbidden. Irritating. Oh, okay. since it was used in a previous study that was effective, then we probably don't need to worry about it. But we still should keep in mind that it is a very small sample. But still, a really cool article. I had no idea what a microglia even was before I read this. I thought this was a really, really cool thing to see. Might look up the guy who made it, see if he has something else that's cool. But yeah, that's it for this video. Thanks for listening to me ramble. I might put some, like, I don't know, fireplace sound in the background. And if you come across this video, for whatever reason, I'm just going to treat it like a journal. Go ahead and leave a comment and tell me if there's anything I need to change, like microphone settings. Maybe I'm talking too softly, or maybe like I'm too close to the mic and you can hear my mouth, which makes some people uncomfortable. Maybe I need to set it farther away. I don't know. If you have anything you'd like to say about the video, please talk to me about it. I'd love to hear critique.